All right. So last part of this, data analysis or sources of information on diet and environment, right? You cannot exclude consideration of environment with the species living on the landscape. So this one, skeletal and dental data. In the image here, again, up close photograph of a tooth with some nice dental plaque attached. So there are multiple options for analysis of bone or dental remains. We're looking at dental first. We noted that teeth are a wealth of information, not just things that might possibly be still stuck on the teeth, but other aspects, for instance, of health. Here, this individual had bacterial decay on the bone of the upper jaw, of the maxilla here, and the mandible as well, although all the teeth are present. Some of these other lines and so forth are also part of the data that we can collect or identify. We're going to look at a couple types of chemical analysis of teeth and bone and look at how they give us extra information, not just what was eaten, but where the person was when they were eating or drinking those materials, and sometimes a record of stages during their life when it's not just calorie interruption, it might be nutrition interruption, and then sometimes cycles of illness. In this case, we see that looking at skeletal and dental information is telling us that there's bacterial infection in the mouth. So lots of information about lifestyle and so forth. So dental plaque again, just spelling dental plaque, that's the combination of things that can be stuck on the surface. This individual has a little, but not a lot. Sometimes we see much more. So since we're looking at dental first, most of the techniques can be used for bone analysis as well. Okay, so isotopes. In your notes, the acronym IRMS is something that might show up in assessment. So isotope, ratio, mass, spectrometry. That's what IRMS is. Isotope, ratios, using a mass spectrometer. And we learned previously in regards to understanding how old things are, that isotopes of certain components of the environment, as they decay and settle back down to their stable state, are really of use for archaeology and many other sciences. All right, the three that we're going to look at real quickly here, carbon-13, that's its unstable isotope, or the isotope they're looking at. Carbon-12 is just stable carbon in the environment. Nitrogen-15 and oxygen-18. So I can see my materials here. When you see this interesting icon here in a conversation about isotopes, then the pronunciation of this is it's delta. So delta 13 versus oxygen 12. We don't have a chemistry precondition for the course, but that doesn't mean that understanding some things that a chemist specialist puts together isn't a great idea for us in the modern world. And then strontium. So carbon, nitrogen, oxygen, and strontium. So two of these, carbon and nitrogen. So again, carbon 13. Earlier in the class, you looked at carbon 14. That's for dating. Carbon 13 tells us about diet. And then nitrogen, N15. Very frequently used to answer questions about diet. Consumption of animal products versus dry land animal products versus fish, how old people were when they started moving them to a more solid diet. Questions like that can be explored with these. Oxygen and strontium, okay, delta 18 and strontium 87, is to reconstruct migration patterns. Biggest use for those and a huge amount of interest is to answer the question, is the person that we found in this location someone who lived in this location their entire lives? Did they grow up someplace else? Or in some cases, when did they move? How long have they been in this location versus someplace else? Okay. C and N, diet sources. O and S are migration patterns relating to what they were eating and drinking, but this is what we primarily use those for. So let's look a little bit more on where. And I will make this bigger for you right now. Easier for you to take a look at. Oxygen isotope in tissue actually gets into your bone or dental tissue because it's present in the water. So going in the water, drinking water, or the food, right? The plants and animals that drink that water and pull it into their system, or their roots pull that into the system. So that value, those ratios are ingested at the time that that tissue is forming. And usually the water in a particular location is closely related to what's going on, meteoric, right? Weather patterns, and will vary over time and in certain locations. So knowing that, taking up things that are the water that you drink or the things that you're consuming that drink water, and then you also drinking some, is going to reflect what's being used to build your system. So most important with these during childhood, sometimes still during adulthood though, there are some things that will change or can modify as an adult. So in this case, for the oxygen isotopes, animal and human teeth and bone. So that delta 18 is a really strong tool if that's something we can recover, because your body chemistry preserves the oxygen ratio, the delta 18 ratio of the rainfall, and therefore we can help to identify migrants and potentially where they actually migrated in. So one of the things that we can reconstruct then sometimes in human history is analysis that lead to the understanding that in lots of areas, biomales grew up and stayed with their families in the zone, town, valley, region where the families were, but their brides came from elsewhere, that they were making connections or some sort of kind of social and political associations with people from further away. And And then, so if you were born a biofemale, then you would not expect that you were going to live with your biological family and close neighbor friends for your entire life. So the reconstruction not only says some of those things about the changes, but things like marriage patterns occasionally, or trade and travel. So reconstruction of that human environment interaction, sociocultural decisions, and individual life histories. So if it looks like most of the brides are coming from elsewhere, then this is the whole societal practice. Sometimes we just see one individual, though, that has a signature that's not local, and we're looking at individuals. So what we're doing also is then recreating their ancient climate and seasonal patterns. This can be done from animal samples as well. So what are some of the practices of trading or growing or seasonally taking your animals from one place to another? Some of you are aware it's not as many people now, but until really recently, in not too many decades, there were huge groups of people taking massive herds of animals, really long distances every single year, up and down at altitude, or to get out of a freezing area and down to a place where it wasn't too cold, or searching for water or food or so forth. So this has been something that we would like to learn more about from our ancestors using isotope analysis. Even cultural practices such as breastfeeding, when are they weaned, and how is food prepared as part of this. And then the human and animal lifetime mobility. And again, that word always important in archaeology, provenience, knowing where things come from. Okay, so this side, just several different ways of mentioning the variety of information we get when we can get this data from isotopes. How about strontium? Strontium's a big one now. It's one of the ones where you probably hear about it more frequently than almost any other type of analysis. So strontium starts in the soil, then it's sucked up by plants, then it gets into your body. So strontium isotopes. And as you can see from this website, the new hot archaeology trend beginning after about 2010 or so, and still going into the future. So strontium, also found in bone, can also infer geographic location at the time the person was growing, when they were still making bone. So what is it? It's a geological chemical signature that we use in order to discover where people were at certain times during their lives. So strontium is going to get into the soil. Then when plants are taking those things out of the soil, that's why we need plants in our diet, to get minerals in our soil or into our bodies, because we don't eat as many plants as we probably should. Then that gets taken up into the food chain, right, by the plants. So the strontium gets embedded into the organic part of an organism. Strontium, when ingested this way, can replace some of the calcium that occurs in bone and tooth enamel, and that is what the specialists are looking for, the ratio of certain levels of strontium. So again, what's strontium used for? Where an individual has been, where they were for a prolonged period of time. If that might make them stand out from most of the other people in the population. So what big change did these types of isotope analysis bring to archaeology and the understanding that we have of, of people and their lifestyle and their communities. Before we move on to toothware, the biggest change was the ability to stop thinking that everyone in the ancient world didn't go anywhere, that they didn't change valleys, regions, villages, towns, that they only reproduced or had marriage contracts with people that they grew up with or that were local or that spoke the same language. This has really, really made our understanding of humans and the way that humans have lots of options to survive and stay alive and do well through the generations to much more complex. So big changes with isotopes. All right, toothware, as we go through this one. Toothware gives us information that in order to really understand this, we do need to do experiments because we can tell after we run the experiments and say, for instance, have an experiment where you're rubbing certain types of plant material versus certain things that might be leather versus things that could be bony or scaly or super cooked meat versus raw things, and see over time, over a long period of time, is there any difference in the wear and tear or abrasion levels that we see in the teeth? Another one that's not really exactly from chewing or force or rubbing, but that gives us a strong amount of information about dominance of certain things in the diet are cavities. And then plaque analysis, that's a separate one. Plaque, then the accumulation of things on the surface. So they can have phytoliths, right? Those microscopic little plant remains sticking onto the surface of the teeth that gets embedded there because the bacteria and other organic things that are stuck on there are actually hardening over time. And we can also get some chemical signatures from plaque analysis. We can get this from dinosaur teeth, from Ice Age mammal teeth, right? This is not just to understand about humans. So paleontology also uses these techniques. So let's look at tooth wear pattern in a little more detail. So abrasion just means different levels of wear, patterns of how the tissue in the teeth formed, cavities and things like that. So let's look at this one. This is a radiograph, right? So a real basic type 1, nothing super fancy about this. They just went ahead and put it under the scanner. And this is from Utsi, or in English known as the Iceman that you learned about in the first unit in a case study. So they have done all that they can think of to understand who this person is and was during his life because his remains were so incredibly well preserved compared to anything that we found in that region and for that age. And one of the things that really stood out was how worn down and smooth and flattened his teeth were. You can see this here. All of his teeth were really worn and flattened. So something he was chewing on was actually hard enough to wear down his enamel, but it wasn't just the front teeth, it was all of them. And also here with the X's, the Iceman also did not have his third molars, either on the top or the bottom. This is sometimes known as your wisdom tooth, for those of you that haven't had a bioanth or an anatomy class. And this is something that would help link him to some biological profiles of local populations there. Now, once we see this wearing, they're starting to think, was he using his teeth for work? Is he chewing on things like leather? But usually that just wears down the front. Is he eating a lot of grains that were produced on stone tables, right, where you're grinding your grains down to flour? That puts a lot of silica or sand in your food, and that wears the teeth down, and that is highly likely to be one of the major reasons for his flattening. But there can be other things going on. Calculus and plaque. So here's an example. So here's, again, the first one that we looked at. Don't want to give you too many visuals that are different, so that if something pops up, part of assessment, it will ring the bell of what we've all seen together, or that we all saw in the reading. So this is the close-up of a Neanderthal tooth. So we can get this from human ancestors as well. You can see a nice little extra lumpy, scaly stuff here. And it did contain microfossils like phytoliths and things like that, from what that individual had been eating. So one of the big surprises in the analysis here was in the microfossils, the super tiny fossils there from this Neanderthal plaque, was that when they went right then, so A and C, these are there on the top row, was the samples that they actually took off the tooth. B and D are from a library of microfossils of things that we know of, plants, seafoods, right, other kinds of things that we might have in libraries. So they started comparing them. And in this case, with the A microfossil matched identically to B, and this is barley. And why is that important? Some of you already know, because the barley it's a plant, right? It's like wheat, oats, that sort of thing. Because Neanderthals were thought to have mainly subsisted almost entirely only on meats and animal fats, fishes, things like that, eggs. What we found is that they'd eaten frequently enough, maybe not every meal, and certainly not during the coldest part of the winter, but that they were collecting plants and eating them. Most importantly, though, in C and D, this is the microfossil, and here you can see it matches this one from the plant library. This is cooked barley. So now we know for sure from this type of analysis that Neanderthals were cooking their foods, right? So in firm containers with water and other sorts of things, making soups, stews, and other softer ways. When people start cooking their food, which we know people started long before Neanderthals, it just was something that we didn't expect from people in the coldest climate. That then with cooked foods, you're able to wean your babies much younger. When they can eat really softened, cooked, and smushed down foods, then breastfeeding can be terminated earlier, and pregnancy tends to happen sooner when the mom is not still breastfeeding. Now, the term pathology, let's look at skeletal things, but diet in teeth also can do this. A pathology is when we see, let's go back to this term, hypoplasia and a pathology. A pathology is when we see something in teeth or bone that is not the standard. It doesn't mean it's a disease, but it's something different. So if you have a filling, right, which helps your health, then you have a dental pathology. If you've needed medical help to repair a fractured bone so that you have a pin or a plate, something like that, that's a pathology, even though it makes everything work perfectly well and now you're well. Pathologies can be like the Iceman worn down of the tooth. That's a pathology, even though he still had his teeth and they were working fine. So this is just giving you a change from the pure standard. And we can see these, some very easy identifiable ones for the specialists in osteology that are related to on and off dietary insufficiency. And also we can see it in dental enamel as well. So hypo means less, plasia means tissue. So we're going to see that the tissue didn't grow as much as it should have in this example. So go down. There you go. Good picture for you. All right, let's get a little bigger. So hypoplasia is a defect that happens when your enamel is growing, but it has an interruption, right? It didn't grow enough. Usually it's because of malnutrition or disease, but that doesn't have to continue forever, right? There can be seasons for some people where they have almost nothing to eat for months, and then the seasons change and they go back and are able to backfill. So enamel hypoplasia here is identifiable, where we can see a series of lines, see these indents along here. So these are interruptions that happened, not when the teeth were outside and showing, but while the teeth were growing up in the jaws. We can see it on the lower teeth as well. We've also got a little plaque down here on these, and we can see that this person had some gum disease going on as well. So these, we've also can see, right, that there's some wear here. So there's a number of little pathologies here going on, but these, the horizontal lines, this is dental enamel hypoplasia. Now, what do we want you to remember about this? So these changes tell us not just that something was interrupted, but it occurs when people are healthy. When, right, chronically means continuing indefinitely, doesn't come to an end. This is when the teeth resume growing. So we can see that off and on, that the growth is slowed down and then it picks up again. So this is very common in certain areas where we've got seasonal challenges. It can also happen when someone's very ill, where their body's using all their nutrition and energy to repair something or get over an infection or an injury, something like that, and then resumes after that is gone. So this is off and on growth, growing fine and so forth. But once the enamel forms, it doesn't change in this pattern anymore. So this is telling us about what happened when this person was either a baby, if this was their baby teeth, or when we see it in somebody, these teeth emerge or come out, right? They drop down beginning about age 5 to 6. And then with the molars, they come down a little bit later. And then your third molar or wisdom tooth comes down usually about 18. So these are, again, telling us what's going on with that person while they're a child. So we get patterns of childhood things. So here's a couple of more. Here's a real clear one. Dental enamel hypoplasia. So we see that interruption, but it started regrowing. Therefore, the person resumed health. So again, in this case, hypoplasia is a record. Here's the star for your nose. What do we use it for? Dietary or enamel growth interruption. And what does it tell us about childhood while the child's still growing? All right. Can we see growth interruptions in bone as well? Well, I wouldn't ask that question unless it was a yes. Of course, let's look over here. So these are called Harris lines. It's easier to see on the lower one here, but you can see it a little bit here. See the white line here. In a Harris line, coming across this way, same thing, growth interruptions up here as well. Now, bone, you all, bone in newborns, in children, the middle section of the bone starts and then the growth occurs at the ends. So this Harris line formed probably prior to this one down here, but this person is fully adult. The growth cap down here is now fully fused with the rest of this, and this is your tibia, right? This is right, the lower leg. This is your fibula. This would be the tiny bone that comes along to the exterior of your ankle. So in this case, and you don't need to have, right, all this anatomy vocab, but I know some of you are anthro majors and you might need it for something else. So distal means away from the center. so the lower. And so we can see Harris lines in this case. Then there was either calorie or mineral interruption. So, or the person might have been quite ill, where the body diverted energy or nutrition to healing, or they just didn't have enough for this to form really smoothly without um, faster, slower growth interruptions. Okay, so yes. And okay, bone isotope or carbon-13. So do remember carbon-14 was for dating, carbon-13 is for diet. So let's take a look. Now, carbon-13 isotopes are not something that we want to spend part of our budget on for archaeological remains from the whole planet. There's a few types of environments where this is going to give us some good information about where people are getting food, when they change their eating patterns, or when they switch over to certain plants in the diet. But that's not everywhere. So carbon-13 analysis or delta-13 budget is not part of every project. So two really important circumstances are when people live relatively near oceans, and we're interested to see the percentage coming in, or any at all, of getting these ocean species, either fish or shellfish. Even if they're living near the ocean, if they're getting most of their protein in the diet from things coming from the landscape, right, like mammals, birds, and their eggs and so forth, then you're going to get different values. If it's mixed, then we'll see that that delta 13 percentage is kind of in the middle. So there are some interesting studies from Scandinavia, for instance, where we can really see at the point where people living near the coast are blocking the ability of people whose communities are further inland to get to the ocean. So those people over time, because they can't get over to the ocean and do that activity, not that they didn't like it before, but they just aren't getting access to it, then begin to make most of their food only from dry land. And one of the other real interesting ones is seeing whether or not those people who, once that's a time frame for having domestic dogs, are feeding their dogs, because the dogs now are fully domesticated and stay with humans all the time, or whether the dogs are kind of come and go and are still expected to go out and find their own food. And we do have samples where the dogs clearly are eating the remains of fish or marine mammals, right, whales and things like that, that they're bringing in from the ocean. And the last one, in the Americas, in the regions that do the first steps of the changeover from mainly wild foods, seeds and grains, then we also can see the time frame where they make that switch to a diet that's primarily corn or maize. That will be another area where we might want to use carbon-13 isotope analysis.